Hey! So, we're experiencing a pandemic. Yay? On March 11th of this year, the WHO declared COVID-19 as a pandemic. Ever since then, people were encouraged to avoid big groups, wear a mask when in public, and constant hand washing. You're familiar with how the virus spreads and what steps to take to avoid it. But what actually happens on the cellular level when you come into contact with the coronavirus?
This is the replication of the novel coronavirus. This is our main character. The virus goes by SARS-CoV-2, novel coronavirus, or more mysteriously, the rona. Coronaviruses are classified by its use of RNA, round envelope, and crown of spike proteins. Then there are the membrane and envelope proteins. The nucleocapsid protein lies on the RNA. The only goals of viruses are to live and reproduce, doing it by hijacking the processes of cells. Once in an organism, this virus looks for something. This something, as researchers have found, is the ACE2 receptor protein. It's mostly found on the cell walls of arteries, heart, kidneys, intestines, and lungs. Meant to receive the signal that lowers blood pressure, ACE2 now becomes the lock that the spike protein fits into, opening the pathway into the cell. Once the virion is in the cell, its membrane dissolves and the RNA, programmed with the instructions to recreate itself, is on a mission to be translated. And here comes a ribosome.
With as many as 10 million in a cell, a ribosome is a structure that lets the cell make protein after protein.
This positive sense RNA is translated, moving through the ribosome from its 5' to 3' and making new membrane and replicase proteins. Wait, so what's all this sign business? Well, similar to the structure of DNA, RNA is made up of a 5-carbon sugar, phosphate group, and nitrogenous base. In the ribose molecule, the carbons are counted clockwise from the right. This numbering defines the orientation of positive sense RNA from 5' to 3'. On the RNA, replicase starts at 3', moving towards 5', making a complementary negative strand. This negative strand then acts as a template to make more positive sense strands for each new virion. It's kind of confusing. The membrane proteins are sent to the ER for glycosylation, or essentially activation, and then are packaged in their new home with the new RNA strand in the Golgi complex. The new virion buds off of the cell. This process is easily handled by the cell and makes from between 10 to 100 new virions, but this cell's incredible virion building is what leads it to its downfall. The body's inflammatory response eventually overburdens the cell, forcing it into programmed cell death or apoptosis. And as one cell is infected, the number of virions in an organism increases exponentially. The good news, however, is that scientists are finding many solutions to fight COVID-19, including those that target the replication process.
Because of this knowledge of the cell's processes, scientists are one step closer to bringing the world back to normal. Stay healthy.